There's something hiding inside all of us. Deep within our DNA, we harbor the remnants of ancient viruses. These special viruses, called retroviruses, infected the reproductive cells of our ancestors millions of years ago. And through this infection, they entered our DNA and were passed on to us as endogenous retroviruses. Today, these endogenous retroviruses make up over 8% of our DNA. One of these endogenous retroviruses is called endogenous retrovirus K, or ERV-K, and it can produce an important protein called integrase. Integrase functions by breaking our host DNA in order to insert viral DNA, which is actually how these viruses infected our DNA millions of years ago. Through this process, integrase causes DNA damage, and this damage needs to be repaired. Integrase itself cannot repair the damage on its own. Instead, it relies on our host repair mechanisms to recognize the damage and get it fixed. If the damage is fixed, integration of the virus is considered successful. But this isn't always the case. Sometimes DNA damage is left, which can eventually accumulate over time and then eventually lead to cell death. If this were to happen in your brain, for example, it could lead to the death of neurons and eventually neurodegeneration. Fortunately, most of us don't need to worry about this because ERV-K integrase isn't expressed in normal, healthy individuals. But in some disease states, it is expressed. Our lab has found experimental evidence of ERV-K integrase expression in the brain and spinal cord of patients with ALS. My research focuses on this ERV-K integrase protein and whether or not its expression in previously healthy cells leads to the accumulation of DNA damage, inflammation, and cell death. And my goal is to determine if this is what is linking ERV-K integrase to the neurodegeneration in ALS. Currently, we don't know what causes ALS, and patients have a gradual loss of muscle function, which unfortunately leads to their death typically only 2 to 5 years after their diagnosis, and they don't have many drugs available for them. If I can determine a link between ERV-K integrase and the DNA damage and all the neurodegeneration in ALS, we can look at repurposing already FDA-approved integrase inhibitors as new drug options for these patients who are in desperate need of new treatment. Thank you.